So methylene blue has a double action, and nothing else I have found can do that. Not even the near infrared light. It not only accelerates the oxygen consumption, ATP production, so accelerate energy, chemical energy availability. It also reduces oxidative stress by preventing these increases in reactive oxygen species.
Hello, Dr. Gonzalez-Lima. You are a professor at the University of Texas, where you run a lab which investigates neurocognitive enhancement and mental health. So welcome to Modern Healthspan, and thank you so much for joining us today.
Thank you very much for the invitation. It's a pleasure to be here and look forward to answer your questions.
Thank you. So, Dr. Gonzalez-Lima, you have been studying methyleneBlue for a long time, and I do want to get onto some other mechanisms of kind of improving mental health. But I wanted to start with methylene-blue. Now, could you introduce methylene-blue? So what is some of the history and what is the chemistry of it?
Yes. Methylene-blue is a very unique chemical compound because it was the first synthetic chemical in the history of medicine. In other words, methylene-blue was the first synthesized chemical that was used for medical applications. And it started out, I would say, in the late 1800s, after the compound was synthesized by a German chemist. It was first tried out on a live animal by another German scientist called Paul Ehrlich in 1886. He reported that he has injected methylene-blue in a live rat, and after he examined the animal, opening it up to look at the distribution of this blue stain, he was very surprised that it was primarily concentrated on the nerves. That is, the different nerves that are around our body, and in particular in the brain, and also in the retina, that is also part of the nervous tissue. So this property has never been seen. Remember, we're talking about something that happened nearly 140 years ago. And he coined the term in English, magic bullet, to refer to a compound that you inject into the systemic circulation in an animal, and it's supposed to go everywhere and stain more or less uniformly. However, that it goes right and stains the nervous system primarily. Not uniquely, but that's where the stain concentrates. And this is the key for understanding the primary action of methylene-blue that I am interested in. So it is a compound that has affinity for something that goes on selectively in the nervous tissue and to a greater extent than in other tissues in the body. And what that was discovered much later was that it is a compound that can readily exchange electrons when it has very low concentration levels. And these concentrations levels of methylene-blue, as in many compounds, are critical for talking about what are the mechanisms of methylene-blue, because low dose, low concentration of methylene-blue does the opposite to high concentrations of methylene-blue. In other words, it is misleading to talk about methylene-blue does this and that. You always have to qualify it. Very low dose methylene-blue does this, high dose methylene-blue does that. And the primary focus that I have is in the low dose methylene-blue. That's the one that we have found to be beneficial for neurocognitive enhancement and also neuroprotection. The high dose is of no interest to us because it does the opposite. However, it is very misleading in the literature that people then say, oh, methylene-blue does this and this and this and this and this. Low dose methylene-blue, usually they are referring to the higher concentrations. And this has led to misunderstanding in the field, and it's led also to false warnings about what methylene-blue can do because, again, depending on the dose, you have opposite effects. And this is not new in the field of toxicology. The number one rule in toxicology is the poison is in the dose. In other words, it is not the chemical itself what it can become a poison. In the majority of cases, with few exceptions, it is the concentration, it is the amount, the dosing, what can be toxic to an organism.
Thank you. Yeah, and let's come back to the dosing because I think the dosing will be a bit complicated. In terms of the mechanism of action, can we go into a bit more detail on what methylene-blue is doing there and how it's kind of supporting the energetics of the cell?
What happens is what the unique property of methylene-blue that unfortunately I have not been able to find in any other compound that has been synthesized since 140 years ago, is that at very low concentration, it reaches an equilibrium between what is called an oxidized and a reduced form of that chemical. Let me clarify. Oxidation refers to the process of removing electrons from a particular chemical element. So, and reduction, the opposite phenomenon, accepting electrons. So, methylene-blue is referred to of a class of compounds that are called redox compound, reduction oxidation. In other words, that they can engage in moving electrons in and out of other compounds. This is a very general property. What makes methylene-blue unique is two things. When it reaches an equilibrium between the oxidized form, which is the blue form, when methylene-blue is oxidized, it absorbs light of different wavelengths and reflects blue light. And when methylene-blue is reduced, it is clear. It doesn't reflect any light, so you see a clear compound. And at the low concentration, the two reach an equilibrium. And what that means is that methylene-blue takes electrons from surrounding chemicals, but at the same time sends electrons back. So it becomes an electron cycler. As long as it's maintained in that equilibrium, redox equilibrium, it acts as an electron cycler. So when methylene-blue was injected by Paul Ehrlich in the rat that reached the nervous tissue, the reason it was concentrated in the nervous tissue is that the nervous tissue has neurons, and neurons inside have an organelle called mitochondria, which is present in all of these other cells that we have in our body, let's say the human body. However, it is present in larger amounts in neural tissue, in particular in neurons. And the reason mitochondria attracts methylene-blue to concentrate is because inside mitochondria, we have a process that is called the electron transport chain, where there are specialized, very large molecules, not like methylene that are designed to move electrons. And these electrons are moved along a chain of these enzymatic complexes that is called, 1, 2, 3, and 4, until the electron is accepted by oxygen. Oxygen is the ultimate electron acceptor in nature. That's why the process of removing an electron from a chemical is referred to as oxidation. And what methylene does is then exchanges electrons with the electron transport. So it becomes a source, a donor, an electron donor in the electron transport. When that is taken up by oxygen, that process inside the mitochondria is coupled with what is called oxidative phosphorylation. That is the conversion of another molecule called adenosine in a phosphorylated derivative. And adenosine can have one phosphate attached to, so adenosine monophosphate, or 2 adenosine diphosphate, or 3 adenosine triphosphate, or ATP, as is commonly known. In nature, in organisms like us, we use oxygen to generate ATP in the process of aerobic cellular respiration. That is the use of air with oxygen to generate ATP. But what is the purpose of ATP? Why are we dependent on that? As you know, if you stop supplying oxygen to the brain, to the nervous system, it's like you unplug a device that requires electricity. It stops working very quickly and starts atrophying and degenerating, and you die in the process fairly quickly, in a matter of minutes. And what ATP does is that when you break down that last phosphate, boom, it is a reaction that releases heat. Chemical reactions can either require heat in order to proceed. In other words, you have some reactants that are right there next to each other, but nothing happens. You heat it up, and then boom, they become a product. Like you remember in like chemistry lab, you can have something in a beaker and it looks one color, then you heat it up, and then boom, transform in a product that is a different compound now. All of these reactions that we have in a living organism like us tend to be controlled by the release of heat. That is, they are reactions that require that heating up. And when you break that phosphate, it releases heat. This is what we call in common language calories. So the amount of food that we eat has to be chemically processed into an electron donor molecule that donates its electron to the electron transport to generate more ATP through oxidative phosphorylation, so that when we break that third phosphate, it releases heat, calorie. And when that happens, some reaction makes reactants here into a product. And that's how we control everything chemically that happens in the tissues of our living body. So that's why we refer to ATP as the energy, the chemical energy required for us to do anything. We have to synthesize these compounds that we want to work with, but they're not going to become a product unless we provide the heat for that reaction to happen. So methylene blue does something like the food that we eat that is used for energy purposes without us having to eat more. It readily feeds electrons to the electron transport. It facilitates the formation of ATP, and then you have more energy resources. And it is like if I give you more money and you go to the shop, you can buy more things. So that's exactly the analogy in the brain. When you have more energy resources, you can do things better because of that. So this is the primary chemical mechanism that I'm interested. And in the low dose, with the equilibrium between reduced and oxidized forms of methylene that's the one that is benefiting this process. This process has different names. Overall, cellular respiration, more accurately, mitochondrial respiration, and is a process that is the essence of life as we know it, that is life that depends on the availability of oxygen.
Can we just
So
go into
that's what methylene does.
Yeah, so one thing in there, so methylene improves the usage of oxygen, or it replaces it in some way?
What it does is facilitate the movement of electrons towards oxygen. So when you move more electrons towards oxygen, more of the oxygen becomes changed. We use the term in chemistry, reduce into water. So you can consume more oxygen when you have more electrons going through the electron transport chain. And the last step is this enzyme called cytochrome C oxidase, or just cytochrome oxidase, that is the one that catalyzes the reaction of oxygen becoming water. So in a way, it improves what normally happens in that process, because one of the reasons that the use of oxygen to generate energy can have a undesirable side effect is that if you send a large amount of oxygen to a particular tissue that is demanding more oxygen, the amount of oxygen can exceed the capacity of that electron transport, and the rate limiting enzyme, the choke point, is that cytochrome oxidase. In other words, cytochrome oxidase cannot make more oxygen into water. And then that oxygen that is not fully reduced to water, that is an inert compound from the point of view of these redox changes, then becomes a reactive oxygen species that is also known as an oxygen-free radical, and that this is the phenomenon that is referred to as oxidative stress. When we accelerate the activity of oxygen consumption and there is not enough enzyme to keep up with it, a fraction of that oxygen will not turn into water and will become an oxygen-free radical. Superoxide is the name for the first step. And that one, because it has an unpaired electron, it will try to share that electron with another compound inside the mitochondria, elsewhere in the cell. And when it does that, it produces what is called oxidative damage. So methylene has a double action, and nothing else I have found can do that. Not even the near infrared light. It not only accelerates the oxygen consumption, ATP production, so accelerate energy, chemical energy availability. It also reduces oxidative stress by preventing these increases in reactive oxygen species. So some people out there say, well, you can live longer if you eat less. Because if you eat less, well, you have less calories. In other words, less electron donors that are working to send their electrons to oxygen, and therefore there is less oxygen consumption, but along with it, there is less oxidative stress. But yes, you do that at the expense of having less energy, less ATP production. So yes, you may have less oxidative stress, but you also have less energy to do things like I told you, all of these biochemicals reactions. So methylene doesn't do that. So lowering your oxidative stress is enough to extend lifespan. However, if you do it with methylene, you're not only extending lifespan, but you are increasing health span because you're going to have more energy at your disposal, especially in the nervous tissue. All of the tissues in the body benefit from having oxygen and from this process. It's just that the nervous system is the one that is more dependent on it. It doesn't have other alternate ways of obtaining energy. It really depends on air from the point of view of neuron action. There are all the cells in the nervous system, like the ones that are called glia, that they can obtain their energy in a process that doesn't require oxygen, so-called anaerobic, as opposed to aerobic. Aerobic meaning air with oxygen, and anaerobic, not air, not necessary to use oxygen. But neurons are highly dependent on this process. So methylene helps on the longevity and neuroprotection by decreasing oxidative stress, which in turn decreases oxidative damage, but it helps to improve, for example, neurocognitive functions or brain functions more generally, because it just delivers more energy to the neurons. And they are, whatever they're trying to accomplish, they can do better. And we know for sure that mitochondria and their capacity for this mitochondrial respiration is crucial for the lifespan of organisms that depend on oxygen, like us. So one of the major theories of aging has to do with the mitochondrial theory of aging, and the other one is the so-called free radical theory of aging. Those two things are targeted by methylene.
And just quickly, so methylene blue is different from other kind of antioxidants in that it stops the formation of the ROS rather than quenching it once it's created. Is that correct?
Yes, very good point. Very good point. There are many antioxidant compounds, but they have two big problems. The first one is the one that you mentioned. They're acting not at the very beginning of this chain reaction, because it goes from what the body tries to do is to reduce that reactive oxygen into a species that is closer to water. So it goes from superoxide to hydrogen peroxide and then to hydroxy radical. So it is becoming closer and closer to H2O. And many antioxidants can do that at different stages of this, but not preventing the beginning of the problem, and which is what methylene blue does. That's the number one. And the number two difference is none of these antioxidants really can get into the mitochondria at the point where this electron transport is delivering electrons to oxygen. They will get, the closest thing is to the membranes, the cell membranes or the membranes of the mitochondria, and then they stop, for example, something called lipid peroxidation. That is that these free radicals are trying to exchange their electrons with lipids that form part of the lipid membranes and producing oxidative damage there. And that's where it acts. For example, vitamin C, ascorbic acid, or some of the other ascorbate salts is a very effective agent that could be an antioxidant. However, uh, it doesn't get to the brain in any significant amount through the bloodstream. And once it's there, it doesn't really act directly on this mitochondrial respiration. Some organisms have found a way to do this, by the way. And I have been very curious and puzzled on how they can do it. So, for example, there are many species like turtles that are so-called anoxia resistant. In other words, that turtle can get under the water and can sleep under the water for hours without doing any harm to their brain. As you know, if you were to go under the water, we cannot use the oxygen in the water like fish. The turtles cannot do that either. However, they can still maintain neural function. And one of the things, the only major difference in their brains compared to another turtle of the same family, but it's not an anoxia resistant, that is, has resistance to loss of oxygen, is a very high content of ascorbic acid within their brain that is actually synthesized and released directly inside the brain. We cannot synthesize ascorbic acid in our body. We are part of the exception to the majority of, let's say, other vertebrates on the planet that can produce their own ascorbic acid to use it for this, for example, for this antioxidant purpose in the brain and other tissues. So we have to take it from the diet, but it doesn't get to the brain. It gets to other parts of the body and it's excreted. However, that doesn't mean that it's a bad thing to take, for example, a vitamin C. Some people say, oh, well, you can take more vitamin C, but then you're just going to urinate that. Well, you also urinate methylene-blue. You don't want the vitamin C or the methylene-blue to become a structural component of your body. You just want it to perfuse your body and do some chemical effects. The vitamin C circulating through your blood, throughout your body, is actually having a beneficial antioxidant effect. It's just that it doesn't get into the brain. The vitamin C, the ascorbic acid itself, looks very similar to glucose. Glucose is the main nutrient that the nervous tissue uses to take up from the blood in order to create those electron donors that I told you that are replaced by methylene-blue. And it's something called the glucose transporter. Because ascorbic acid is very similar to glucose, that transporter has always preference for glucose and not for ascorbic acid, and then ascorbic acid cannot get into our brains in any significant amount. If we could find a way to do that, like these anoxid-resistant turtles and other animals like that, I mean, people wouldn't have strokes. People could be swimming under the water for hours. We will have, but that is not possible. So our best shot, at least that I have found, is still methylene-blue. And I believe now that there is a lot of evidence that in many of these anoxia situations or hypoxia, anoxia means just without oxygen, and hypoxia, low levels of oxygen, we can demonstrate the beneficial action of methylene-blue not just in the brain, but all the tissues that require loss of oxygen, like, for example, your heart. One of the reasons you have a heart attack is a reduction in the amount of oxygen reaching the part of your heart. Well, you can do this experimentally, for example, with pigs. It's been done a number of times. And if methylene is on board, you can significantly prevent the damage from to the heart. And also you prevent the damage to the brain because when you have a heart attack, you also compromise circulation to your brain, which delivers oxygen to your brain. So again, the end result is similar. So this is one of the reasons why methylene is so unique.
Yeah, that's really interesting. So actually talking about preclinical and clinical data. So what have we seen in terms of preclinical? So mostly what you're looking at is, Alzheimer's and cognitive impairment or cognitive behavior. So what have we seen in animal models for using methylene blue and cognitive decline?
Sure. Well, it is very important to preamble that by saying the in vitro effects of methylene blue are not necessarily relevant to in vivo effects. That is, the effects that happen in a petri dish or a test tube where you put methylene blue may have nothing to do with what happens in vivo. Because in vivo, you need the circulation, you need to be breathing, you need oxygen to be delivered to the tissue. Not these things are happening there. So one of the biggest, and perhaps the biggest misconception problem with the use of methylene blue or, for example, dementia, Alzheimer's disease, has been the finding that in vitro, methylene blue, because of its redox properties, can prevent the aggregation of one protein that is called a tau protein. And this tau protein aggregates in large bundles in the brain of many degenerative diseases, not just Alzheimer's disease. Once you see this tau accumulation inside neurons in these neurodegenerative diseases, means that neuron is functionally dead. It doesn't matter if you remove that tau from there, that neuron already lost its functional capacity. So it is a consequence of a process of neurodegeneration to have this built up. So in vitro, you add, the more methylene blue you add, the less of this tau aggregation that happens. So there was a group that found this in vitro effect, and then they jumped from that in vitro effect to saying, oh, if we give it to patients with Alzheimer's, that they have this accumulation of this tau on those neurons, we're going to be able to rescue them by interfering with this tau aggregation. And the more, the better, because in vitro, that's what you get. However, like I just explained, the really valuable benefit of methylene blue comes from the low concentration redox equilibrium, reduced, that makes it an electron cycler to mitochondria, a process that is unrelated to this tau phenomenon. So they tested people using these large amounts, large doses of methylene blue, and essentially they were not able to find benefits. And they also did this with animals, with primates, monkeys, and they would give larger and larger amounts, thinking that this would be better and better. And the results always pointed to the opposite, that the so-called comparison or control, quote unquote, placebo group was actually a very low amount of methylene and the one they expected to work was the higher amount, because this very low amount doesn't do much at all to this tau aggregation in vivo or in vitro, for that matter. But these ones were the ones who benefit more than the other ones. And they came up with all kinds of explanations for this and never accepted the fact that the whole premise for the company is to eliminate tau aggregation. So they have never acknowledged these metabolic effects of methylene being the ones that are actually, you don't need to have Alzheimer's disease in order to benefit from methylene because everyone that has a nervous system that is able to use oxygen and you are alive will be able to benefit from methylene. So this is not something specific like what they were trying to do, link to neurodegeneration. So what we're doing is simply providing more of these energy resources and diminishing the oxidative stress that goes along with producing more energy. And when you have that, and then you put somebody in a cognitive task, an animal or a human, then they have more resources to do that task. You can also recreate a situation that will produce degeneration in an animal under very controlled conditions. Like I mentioned to you, you can diminish the amount of blood flow that is going through the carotids to the brain, which will reduce the amount of oxygen. That will be the most immediate thing. Of course, you reduce the amount of blood generally with other nutrients and chemicals. But if you have methylene on board, you can prevent then this neurodegeneration that results from the loss of the blood supply, the oxygenated blood getting into your head, into your brain. And you test the animals that do not get methylene they get another blue dye. So people don't know which animals are getting what. These control animals do very poorly in this memory and other cognitive tests. And then they show neurodegenerations when you examine their brain. And the ones that get methylene at very low concentrations are able to prevent that. We call it a neuroprotective action. You can test them. They don't show these deficits in memory. You can look for neurodegenerative changes and you don't see them in their brains. So these are some of the studies that our group has done, and primarily with methylene starting out with the retina, that we could demonstrate that you could prevent a degeneration of the retina induced by a toxin that affects mitochondrial respiration. You can prevent entirely, or putting in a region of the brain, for example, the striatum. It's a region that is related to Parkinson's disease, the nigrostriatal pathway. You can put the methylene and that prevents that region from degenerating out, and the animals continue to show normal movements. So all of this has been done under very well-controlled conditions in animals. In humans, it is more difficult to control things. And the studies that were done, like I mentioned before, were done by a group that misunderstood that the important thing was the low dose pharmaceutical-grade methylene for its redox properties that facilitate mitochondrial respiration, and not something related to tau aggregation. And therefore, the literature is confounded. We knew from the beginning when this group was going to do this, and a trainee of mine that was consulted by them, they told him that all of this was going to happen but that didn't make any difference to them. And I had to say, I review many of their papers. I pointed out all of these things and more. Didn't change a bit. Uh, they had a very simplistic understanding. In vitro, it stops aggregation of tau. So in vivo, it must do the same thing, and therefore it can prevent these neurodegeneration changes. And that doesn't happen. So
Have
methylene
you?
blue
Mhm.
doesn't require any kind of amyloid or tau things that are happening in some of these neurodegenerative diseases. Doesn't require any of that to allow its benefits to take place because it's acting on these other phenomena of mitochondrial respiration.
Have you done any studies on age-related decline in either mice or humans? And
Yes,
also,
in animals we have done it. Yeah, you can prevent these changes in animals, and especially if you combine an animal model, for example, in a rat, that is more similar to what happens in us humans. In us humans, you have two things are acting. When we're getting older, we're not only having a problem with the mitochondrial respiration itself, uh, and we're building up this oxidative damage from the cumulative effects of our lifespan. We are also having a less efficient vascular supply to our brain. So our cardiovascular system declines year by year after mid-age, our middle age. So we have less oxygenated blood that can access our brain. So in animals, we reproduce this combination because the two ingredients are important. We have and there is a third ingredient that I will mention in a moment, but these two ingredients are important because it's not just age, it's also the blood supply to the brain that is being slowly compromised. So we call it chronic hypoperfusion, cerebral hypoperfusion, less perfusion with oxygenated blood. So in those animals, you give methylene blue and you prevent the problems that are happening in the control animals. And in particular, we do this starting out with animals that are, for example, middle age or older to show that we can prevent things that are going to continue to be aggravated when you do this. There are some experiments with very simple animals that are less compatible with humans, it's like some flies or animals like that. And methylene blue in those animals can extend their lifespan. It is easy to measure. You can see it without a problem because they have very little lifespan. Uh, it You can also do memory tests in fish. I have done that in little fish. Uh, you can demonstrate that this fish will show better memory if you put them in a container that has some methylene blue as opposed to blue food dye that looks the same. You just need to put it there for an hour or so and then put them back, and they are able to retrieve the memories better, where to find the food, how to move around, if you have trained them for that. So this is a very general phenomenon that can be seen. If the animal has mitochondria, and the mitochondria are the major source of energy in their brains, then they benefit from this. But we do need more studies in humans, and that's why I plan to do one. But I don't want to do just methylene blue. I want to combine it with near infrared light because methylene blue, you have to keep it at a low dose. So as the problem aggravates when people are older and have more cognitive decline, you just cannot have more methylene blue on board. And there is a limitation of how far you can improve this mitochondrial respiration as well. So I found another way to do that with this near-infrared light targeting in a different way, but targeting the same mitochondrial respiration mechanism.
Okay, before we dive into the red light and methylene blue together, let's talk about dose, because you've referred to dose all the way through, that it has to be just the right dose.
Yes.
So how do we get the right dose? And also, methylene blue, is it, if you take it orally, does it get absorbed? What kind of dose? If somebody really wanted to try methylene blue as a supplement,
Yes.
it's available over the counter. So what kind of dosing and how would you determine that?
Yes. The answer to that question is not complete. In other words, no one has tried all possible doses of methylene blue in animals or humans. But methylene blue is very well studied in humans. Methylene blue has been used in humans for over 100 years. So there is, we know very accurately things like the so-called bioavailability, that is how much methylene blue goes into our brain or our circulation once we take it. An oral amount intake of methylene blue actually works better than injecting it directly in the circulation. The reason is when you inject it directly into the circulation, you are creating a larger concentration locally there where you are injecting methylene blue. And you don't want that. You want a slower absorption where you always keep the concentration levels slower. And the primary target when you inject it immediately is the red blood cells, is the cells in the blood, is the blood itself. And that's not the purpose when you're using it for neurocognitive enhancement, for example. That is the most common acute use of methylene blue nowadays, is using it in emergency rooms when you have a problem with any chemical that interfere with the transport of oxygen by your red blood cells. These are the cells that transport your oxygen. They open up, they have a little iron here that captures the oxygen closer up. And when they reach the capillaries, for example, in the brain or other tissues, there is another molecule, cytochrome oxidase, that is open up and has also the same iron compound. It's called a heme. And then it captures the oxygen from the red blood cells. So that's how the oxygen goes from the blood to the tissues. Methylene blue, it is better absorbed to act on that mitochondria, on that phase, as opposed to act on the transport. But on the transport is a lifesaver. That's why methylene blue is listed by the World Health Organization among one of the 100 most important medications in the world that should be available in every hospital. Because if, for example, you get carbon monoxide exposure from the exhaust of cars, and these will bind. Carbon monoxide is similar chemically to a molecule of oxygen that has the two oxygen. Well, it has a carbon and one oxygen, and then it doesn't allow the oxygen to be transported effectively, and you effectively will die from hypoxia. So they inject methylene blue in emergency rooms, and this competes with this carbon monoxide and displaces that out, and then oxygen occupies its natural position in the hemoglobin the molecule that carries it in the red blood cells, and then it becomes oxygenated hemoglobin. That is the one that carries the oxygen, opposed to the deoxygenated one. So the most classic poisons in the history of the world operate in that way. For example, cyanide. From historical time, they know if you ingest cyanide, you die very quickly. Like, I always say, like this Russian spies that carried cyanide in their rings, and when they are trapped or discovered, they go take it and boom, and die. So if you give them an injection of methyleneBlue, you rescue them from dying. The methyleneBlue displaces the cyanide. The cyanide, the important location where it is in that cytochrome oxidase. That cytochrome oxidase cyanide is called a competitive inhibitor of that enzyme. It will occupy that binding site where the oxygen is supposed to be, and then oxygen cannot be processed into water and get the energy, and you die from that. So methyleneBlue is the drug of choice to rescue, and many other examples. Essentially, if you have a metabolic poison that is affecting your oxygen transport or consumption, just about the only thing out there available in emergency room is to give you methyleneBlue. So emergency medicine physicians are very familiar with methyleneBlue for decades and decades, but they have used it only on this acute way. And I told you about the problem with the transport of oxygen is called methemoglobinemia. That can happen when you have some of these poisons that interfere with the transport in the hemoglobin. There are some people that have hereditary methemoglobinemia. In other words, the red blood cells are not effective at transporting this. They take methyleneBlue all their life. They have a chronic oral administration of this, and that's how they can live, because the methyleneBlue is reducing the methemoglobinemia. However, methyleneBlue, if you give in a high concentration, it causes methemoglobinemia, because in a high concentration, it will occupy that space, not just simply displace what is there and allow oxygen that has more affinity to go there. It will compete with the oxygen at a high concentration and will cause methemoglobinemia itself. So this is a very clear example. The same compound that is used to rescue people and save life for methemoglobinemia caused by metabolic poison can induce it if you use it at a high concentration. Lots of misconceptions about referring to methyleneBlue actions using the higher doses. For example, I get asked all the time about nitric oxide. Oh, I read methyleneBlue is a nitric oxide inhibitor. MethyleneBlue at high concentrations will inhibit any of these, what we call oxidases, tissue oxidases. It's just going to compete with them. But at the low doses that are beneficial for animals and humans, it doesn't do that. So there is no concern about that. But they get confused because they only talk about methyleneBlue does this, as opposed to a low dose and a high dose doing opposite effect, just like in the example I gave you about the rescuing from methemoglobinemia and inducing methemoglobinemia. So what are the low doses? Low doses that we have tested. We haven't tested all the possible doses, but the ones that work better are always between 1 and 4 milligrams per kilograms. There, some lower doses, we have only tested very little, but they also seem to work. In other words, is this effect that we refer to as hormetic is like a inverted U curve. So with the low doses, you're working on this part of the curve. That is, as you increase the dose, you have these beneficial effects of methylene but then you can keep increasing the dose until you reach an optimal amount. And then if you keep increasing, it does the opposite. And with methemoglobinemia in the blood, you can do this readily, can demonstrate this phenomenon. But so we have been working only with this linear increase here, but then it becomes this biphasic response. So as long as you remain there, and anything that we have done lower than 5 milligrams per kilogram seems to still be in that beneficial increase. For example, in mitochondrial respiration, oxygen consumption, reduction of antioxidant effects, reduction of oxidative stress. It is likely that any dose that is even 10 times less than 1 milligram per kilogram will still be in that part of the curve. In other words, you can go down without any risk, but you cannot keep going up because then you start, you're going back to baseline and then you're going opposite effects. And this is something that needs to be understood. So low dose methylene and it has to be pharmaceutical grade. The term clinical grade doesn't have really a meaning. It's not meaningful. You have methylene that is industrial grade, that is used for stain, like blue jeans, clothes. At the beginning, all of the blue jeans in the world were stained with methylene. So who knows? We may be absorbing some methylene from early days in very low amounts, because you know blue jean loses these stains as you watch it. Then you have what are called chemical purity or chemical grade that is used in labs or chemistry, but it has a very high impurity levels. In the most common products that are sold, I'm not going to name the suppliers, about 15% of the chemical is not methylene and is chemicals that are actually products of the synthetic process that are toxic, that are chemicals that you don't want to take. So the so-called pharmaceutical methylene reaches 99.99 purity. And that's the one that you want to get. It is the one that it should be used in animals and humans. There are many studies in animals done with the chemical grade, and those results are invalid because they are giving an animal 15% of toxic compounds along with the methylene and sometimes chronically. And I have reviewed many of these papers pointing this out, and these papers continue to be published. You cannot just buy in every pet store. You have chemical grade methylene that is used to put into the water in fish if they have any kind of problems outside their body, like some parasites or things like that. They put drops of methylene in the water, and that's enough to kill the parasites, and the animals are healthier, the fish are healthier. Well, you don't want to use that methylene. Only in the US, the pharmaceutical grade is called USP, United States Pharmacopoeia. There is an European pharmacopoeia as well. However, the requirements for the impurities in the European standard are not as high standards as the one in the US pharmacopoeia. Oftentimes it's the opposite, but in the case of methylene uh, you're going to get a more pure, less contaminated compound when you use the USP than when you use the European. And that creates also some problems with the use of European methylene blue for studies that because the reason is historically in Europe, they were using it like the original to kill these malaria parasites. So, for example, to give you an example, for killing malaria parasites that is still being used, especially in Africa, uh, and especially because of European physicians that go there and treat particularly children that who are really affected with malaria parasites, they give them very higher concentrations of methylene blue. But what happens, they give it them orally. So that higher concentration of methylene blue, that is several times what I'm referring to you between 1 and 4, it gets into the digestive system, into the intestines, and it helps kill the parasites that they have, and very little is actually absorbed through the circulation. The little that is absorbed is beneficial, and the majority that is in the intestine is what actually helps to kill the malaria parasite. And the main reason for this is that the malaria parasite contains an enzyme that is not present in humans that is very susceptible to methylene blue, to be inhibited by methylene blue. So you weaken the parasite. It doesn't really kill it. Your body is the one that eventually kills the parasite, but you weaken the parasite so your body can actually kill it through your immune responses. And so this is how it was first used as a medication. And I can tell you, for example, that during World War II, all of the Navy command under Eisenhower, all of these sailors were taking methylene blue every day as an anti-malaria agent. And they would have songs and things saying that they were so much sailors that they were even peeing blue, uh, which is the major problem with methylene blue. Methylene blue, most of it just gets eliminated through the urine, your urinary tract. And that's why, but this is also very useful for dosing. So if you ask me what's the best dose for a person, I would say it is the best dose for that personalized individual. And you can tell that to a large degree, excluding some of these exceptional situations with some enzymes, rare cases that prevent the use of methylene blue and many other drugs. If, for example, you take the pill and you notice your urine starts discolorating, and I use the word discolorating because if your urine is very clear, it's going to look more bluish. But if your urine is not, you're not so well hydrated, your urine is normally more yellow, then it's going to look more like greenish. It's just a combination of the blue and the green, and the blue and the yellow making green. So you take the methylene blue and you see how long are you peeing with a discolored urine. And this, of course, as you grow older, your urinary system, your kidneys, do not work as effectively at excreting compounds. So actually, older people have their methylene blue circulating longer. They actually can consume less, not more, of methylene blue, like was done with the Alzheimer's studies that they were giving them very large amount. No, because we, as we grow older, have less efficient in terms of urinary excretion. You just have to take it and see how long it takes. If you take it and it takes 5 days when you no longer see any visible sign, and it's okay, just the visibility is enough to see it, then you know that you can take the next dose because you have excreted what you have. If your dose is so small that you don't see any change in your urine, especially if you are well hydrated and your urine is very clear, you should be able to see if there is any of the methylene stain. When we start urinating, as soon as the methylene blue is going out and exposed to the air, it becomes oxidized into blue. When it's in the body, like in your urinary bladder, it's primarily in the reduced form. And when it's in the mitochondria inside your tissues, it's in an equilibrium between reduced and oxidized forms. So when you urinate is when the air oxidizes, turns blue, and it starts staining. So you can personalize this to you. How much do you need? Well, you need as much for you to be able to see some urine discoloration. And for how long? The duration of action, for how long as you see that discoloration. As soon as you start seeing or it's difficult to see, you can take the next dose. And somebody like me, that I'm going to be in less than 2 months, I'm going to be 70 years old, I can only take it once a week is enough because I will slowly dispose of it. And as long as I'm not excreting it, it's still circulating in my blood and having affinity for my brain mitochondria, and I'm benefiting from it. So you don't have to take it every day. You have to personalize it to your own system. And that's the best advice. So one size doesn't fit all. It's what nowadays they're referring to as personalized medicine or precision medicine. But because of this property that you can see the coloring, you can actually, it's like doing a lab test with your urine. You can see the results of what you have taken. And this is very useful for practical purposes.
Okay, so you could start. So I'm about 66 kilograms. So it'd be about 70 milligrams would be about.
Yes.
Yeah,
Or you can
right.
start with half that amount and see if you see discoloration in urine. As long as you see discoloration, the methylene blue in those lower doses will be in that beneficial part of the curve. So as long as you see discoloration and it's less than 5 milligrams per kilogram. So one is 5 times less than that dose. We have done experiments in humans where we give them 4 milligrams per kilogram, but in those experiments, we were trying to look for an acute effect. Like we were going to give it to them now, and an hour later, that we know is passed orally, taking orally as passed to the circulation, perfused in the brain. We were going to examine their brains with fMRI and see how their brain were responding to these, give them memory tests, cognitive tests, see the benefit of these. And this could be done, and we've done it in a controlled fashion because we asked them to urinate before they go to the scans. They take the methylene blue, so they don't know whether what they taken is going to discolorate the urine until after they're through with the acute experiment. The problem with the discoloration for control studies is when they have this, chronic administration every day with these high doses that they have very clear discoloration. It's just too obvious. And of course, if there's going to be discoloration of your urine, you should use underwear undergarment that is dark black, deep blue, because it will stain. It's a stain and it will stain. And even though we don't think so, we do overnight leak some urine. In the US, this is the last point about this. In the US, methyleneBlue was available for many decades. When I started human research with methyleneBlue, it was still available over the counter in a compound that was called Urine Blue, a medicine that no longer exists, that was used for urinary tract infections. And for urinary tract infections, what they wanted was you take enough methyleneBlue that you would reach a high concentration inside your bladder to be a pro-oxidant. In other words, be the opposite of antioxidant. And then without having to use antibiotics, kill that bacteria or viruses that we're producing. And this was very effective, especially for older people, and in particular, older women that had a tendency to have recurrent urinary tract infections because of their slow way, because their urine just stays too long in the bladder. And the dose that they will recommend was 3 capsules of 65 milligrams each every day. So that, as you can see, that starts, if you do that, you're going to start reaching into the other side of the curve. You're going to start taking about 3 times what I was telling you was the 1 milligram per kilogram dose, and you were taking it every day. So that was used very effectively but physicians were trained to use antibiotics, taught in medical school. I taught medical students for 10 years. They are trained to just use antibiotics for infectious diseases that are linked to bacteria, and they abandon the use of methyleneBlue. And of course, this was over the counter, so you didn't even have to go to see a physician, so it was not good business. You needed a prescription of an antibiotic in this part of the world. So, however, these are very debilitating to older people to go through these rounds of antibiotics that also affect the intestinal flora, what nowadays they call the microbiota. And after a couple of months, you may go back and have a recurring infection. So this became a continuous problem, and people were weakened, especially the older ladies, by having to go through this process. So having mentioned that, it is a good point to say the low amounts that we take of methylene do not affect the microbiota. There's been lots of people that have been saying this. I'm sorry to tell you, some of them, the gastroenterologists, working on microbiota based on speculations and anecdotal observations. These low amounts do not affect the microbiota. It has been examined experimentally. Finally, last year came a well-controlled study, and even very high doses, 10, 20, 100 times more than the doses that I'm telling you, in animals where you control what they eat and their environment, do not have an impact on the microbiota. So for whatever reason, this is a myth that has been spreading there. They say, oh, I don't want to take methylene because it may affect the microbiota. The only available experimental results are those. And again, in humans, it's very difficult to do something as accurate as that study that was done in animals, and use all of these very large range of doses. The only thing that happened with those rodents was they improved their memory in their tests that they were being given at the lower dose range that they were doing using methylene.
Okay.
So that was the only positive finding from that. So I hope this helps. And if you have any other questions.
Yeah. So if you have time, because you are now looking at red light and combining red light with methylene blue.
For the most part, I have not done the combination. I'm going to do the combination with this clinical trial that I already submitted for IRB approval. But the benefit of the near infrared light is that you can do a more local targeting. So if you use with a laser or LED over your forehead, that light is going to penetrate and it's going to target your prefrontal cortex. Methylene blue will go to your entire nervous system. So the thing is that as we grow older, the more obvious type of memory that starts to decline is this so-called working memory. That is a shorter type of memory. Like, for example, you go out of your house and then you wonder, oh, did I lock or not? I don't recall. So that working memory is the one that allows you to have this information available. So this involves a lot of prefrontal function that we use for anticipating and retrieving these working memories. So we have shown in humans, not only in animals, but also in humans, that infrared light, and I don't use the word red light for humans, because red light is good enough to penetrate in animals that have small heads or in the superficial tissues like the skin, but you need the near infrared in the order of 800s or 1,000 some nanometers of wavelength of light to penetrate, for example, through your forehead, through your skull, and into the layers until you reach the surface of the cerebral cortex in this prefrontal area. And there are many claims out there that if you use these lights with 600 nanometers, for example, that is red light, they're not going to get there in any. In other words, the number of photons that are going to be available to reach the mitochondria and inside the mitochondria to be absorbed by that cytochrome oxidase, that is then use those photons are going to serve the same purpose as these electrons from methylene blue. They're going to push the electrons from cytochrome oxidase into oxygen, and then will create that same process of oxidative phosphorylation. If there are not enough photons that are reaching the inside the mitochondria, it's not going to happen. Cytochrome oxidase is the major photon acceptor inside cells. And that's where the name comes, cytocell chrome color. Color means you absorb some wavelengths and you reflect others. So it gives color to the inside of the cell, and it's inside mitochondria, in these inner membranes of the mitochondria. So the target is the same, is that cytochrome oxidase, mitochondrial respiration, but now you are providing photons that are able to push the electrons toward oxygen as opposed to donating them through the low concentration methylene blue. And we are going to combine very low dose methylene blue with prefrontal stimulation. We know the two are effective, each one independently. So we don't need to demonstrate that again. And we know the effects of the two in the brains, in humans, in vivo, not just some in vitro extrapolation. We want to put them to work together synergistically. And we're going to add a third element that I mentioned to you at the beginning. One of the problems that we have as we grow older is that the glucose that we normally take in order to generate those electron donors that are going to be able to generate the ATP, it becomes less efficient. That glucose uptake mechanism, it declines as we grow older, becomes less and less efficient. However, we have an alternate secondary route that we use, for example, when we are asleep, when we are not eating in fasting conditions. We have an alternate source that is coming from the liver. The liver is like a little chemist inside our body. It creates these ketone bodies, and these ketone bodies can also be used by the mitochondria, but in a different biochemical pathway that also generates ATP. So when we are in conditions of fasting, and starts out when we're born, like when we're born, we spend most of, if under natural conditions as in mammals, we get all of our energy from the milk, the mother's milk. And that has this high amount of these fats from which you can derive this ketone body. And so we retain that ability that we have of using ketone bodies to generate energy throughout our life, as opposed to the glucose that goes decline. So this is, and it's been finally recognized after a number of clinical trials by the FDA. This is the Food and Drug Administration in the US that gives approval to interventions. The use of ketones or precursors like medium chain triglycerides, a type of fat where your body uses for energy purposes, not to accumulate fat storage. They have been declared a medical food for the management of Alzheimer's disease based on results from studies that have been done. And of course, the main reason that happened was because one company modified one of these medium chain triglycerides to make it a drug, which is a natural compound, but they just added something so they could patent it, and they created a drug. They showed the studies, it works, but you can go to the supermarket and buy MCT oil, medium chain triglyceride oil, and it will do the same as that medication that is. However, they interestingly they created a category for it called medical food, because it's really a food and not a medicine itself, and approved that for the management of Alzheimer's. So we want to combine these three things that facilitate mitochondrial respiration.
And what were the
methylene with the electrons, the photobiomodulation near infrared light with the photons that are targeting cytochrome oxidase, and medium chain triglycerides or ketones. There are a number of derivatives of these ketones that one can intake and also do similar. Of course, the advantage of the ketone is that your liver could be compromised, because you need your liver in order to make ketone bodies from this MCT oil. But in an older person with a liver problem, the ketones themselves, and there are already some pharmaceutical and nutrition companies that are making some of these compounds out there. And so combining the three to maximize then your mitochondrial function and facilitate neurocognitive performance.
Who are the targets for this? Is it Alzheimer's or is it ordinary, like healthy people?
The studies were done with Alzheimer's patients. The ones that were used by the FDA for approval, and that's why they call it a medical food for the management of Alzheimer's disease patients. So these were people who had more severe cognitive decline.
The study that you're planning, who will the participants be?
Oh, the study that I'm planning, I'm going to use mild cognitive impairment, which is the phase that precedes dementia. And because the best use of this is to prevent degeneration to start happening and not to try to rescue the brain once these other problems start happening. And so that's what we tried. We will screen for these, and we will target an older population, about my age, for this intervention. And it should be simple. These medium chain triglycerides or their main ingredients can be also taken as capsules, or you can take the oil itself. So it wouldn't be difficult to administer all of these. The methyleneBlue and the ketones will be taken orally, and then we're going to have people have. We plan to use, instead of laser, which is what I have used primarily for all of my studies, we plan to use LEDs that are available now at the same or very similar parameters of the lasers that we know are effective. Most of the LEDs are sold out there for the purpose of photobiomodulation to improve your cognitive function, do not have sufficient parameters to be effective, unfortunately. And also the ones that they claim that you put it on top of your hair, like with a cap or something like this, the melanin in the hair will absorb most of these photons, and very little, if any, will be able to get in to do any benefit. So most of those things, the only thing they will do is just heat up your hair. Some of those LEDs are called buckets. So the best target is use your forehead. And the melanin in the skin is not a problem. You can have a dark skin. It is the hair, because the hair has all of the filament is filled with it, and you have different densities of hair. It creates a problem with the dosing that is significant. And we don't need it, because what we need is to target the prefrontal, which is the first one that it starts compromising when you start having memory issues. It's just working memory issues that the first one that starts showing up, those are the ones you want to target. And if you have on board the other two elements, you're going to be working with your entire nervous system as well. And so that's the idea. And hopefully, when we see any result of this, will be the PhD dissertation of one of my talented graduate students here. And we'll see what happens. And I'll be happy to come back in a few years to tell the story.
Yeah, that would be really interesting. And it sounds a very exciting study. Would be great to talk about the photobiomodulation, but we've already gone for an hour and a half. So definitely don't have time to dive into that. Okay, definitely looking forward to that study. Okay, so if people want to follow your work in general, where can they go?
I don't really write for the public. As you know, my studies are really published in the scientific literature, but they can go a Google search with my name, and it will show up. Or if they can add methylene blue to the search, It will show up dozens of studies that we have done over the years, and they can then select the menu. There are some other people, like I mentioned, that have summarized some of these in articles more appropriate for communicating to a larger audience. But I haven't done that yet. I have limited myself to there is so much new knowledge that can be gained that we don't want to divert our attention. So what I have done is like with your invitation, talk about this in the hope that people are aware and that, and it has worked. I mean, there is an incredible interest in the use of methyleneBlue all over the world. So I'm very happy to see that. And I hope that some of the other scientists will do more research on it as well.
Thank you. Yes, there is definitely an interest in methylene and I just think the name is not good. I mean, it sounds like a blue dye. So
it is a blue dye.
I know it just doesn't seem like
but
something.
If you add vitamin C to it, it becomes clear. You add some vitamin C to it, and you have it in a closed container. But as soon as there's some air that goes in, it starts turning blue again. So if you don't want to take it that is blue, you just put the vitamin C, which I think is a good idea to combine it with vitamin C, because in order to pass through the membrane, the lipid membranes through our cells and mitochondria, it prefers to travel as in the reduced form. So that normally happens when you ingest it in the stomach, that we have these acids. They will facilitate the creation of reduced methylene-blue. But if you, I always recommend in our studies, we combine the methylene-blue with vitamin C. And so it facilitates the absorption into the body, and it becomes less of a stain.
Interesting. Okay. Anyway, Dr. Gonzalez-Lima, thank you so much for joining us today. It's been great talking to you.
Thank you very much.
Thank you.
Bye-bye.
Okay. Thank you. Yeah. Bye-bye.